Mediterranei a Damola Adejoro no Mayangi Tayao, Rarwa ko Monica Gerth no te whare wananga o te heranga waka. Ko Zwelvanes tenei, ko au kai whakaere mo tenei wananga. So welcome everybody to to the APPS seminar series, Australian Plant Protection Pathology Society. That's seminars that happened every month. Today we have two speakers, Damola Adejoro, who is from the Bioeconomy Science Institute, and Monica Gerth, who is from Victoria University of Wellington. We'll have questions at the end of each presentation, and I would ask people actually to have their microphone and camera turned off, and so that you put all your questions through the chat. So the first speaker today will be Damola. He'll be talking to us about a very interesting and complex relationship between microbiome and trunk disease in grapes. So Damola, thank you very much for accepting the invitation to talk today at the APPS seminar. Perfect. So anytime, Damola, the floor is yours, and looking forward to your presentation.
Thank you very much, Joe. Thank you very much, everyone, for coming today. My name is Damola Adejoro. I'm based in the Bioeconomy Science Institute, Ontario at Hawke's Bay. Today, I'll be talking about the microbiome structure of disease escape grapevines in New Zealand. And this work originated from my PhD at Lincoln University and the Old Plant and Food Research. So I would like to acknowledge the contributions of my supervisors and my advisors. So take a second to have a guess on what this figure means. So this is the total export value from wine from New Zealand last year, $2,098,000,000. And this value, over 85% of these in volume, was dominated by the Sauvignon Blanc cultivar. However, for this industry to continue to be profitable, we need healthy, productive vines year after year. But a big problem affects the continued profitability of this industry, and that is the grapevine trunk disease. So the grapevine trunk disease is used as a collective term to refer to different diseases that affect grapevine trunk. In New Zealand, the common ones are the Phytophthora dieback and the Utipa dieback. So they manifest as cankers in trunks and also stunted vine with chlorotic shoots. And also there is this characteristic wedge-shaped lesion seen in the cross-section of the vine. One of the reasons that makes this disease complex is because there's a lot of pathogens involved in their etiology. And in New Zealand, from work carried out by Dion and colleagues, we've seen that Utipa lata is a very common trunk disease pathogen in New Zealand, as well as Pneumococcal and Pheomonella, Caduphora, and a host of others. And this is why it's a little bit complex than say your regular disease, because so many organisms are involved. Remember, I said in the opening slide that Sauvignon Blanc constitute over 85% in volume of wine exported from New Zealand. It is actually one of the most susceptible cultivars to trunk disease, according to work out of Australia by Max Sosnowski. In this research, they found out that Sauvignon Blanc has the highest severity to trunk disease pathogens. And this trunk disease leads to reduction in yield because there's no productivity from this trees. And it increased management costs because the growers need to replace vines, prune trees and treat them, and also remove the infected wood from the vineyard. And currently, we don't have complete eradication. The control strategies we have for the trunk disease include the protection of perennial wounds and sanitation by removal of the infected materials from the vineyard. And also, if all else fails, there is a remedial surgery where we can cut down the infected tree until the wood staining associated with trunk disease is no longer visible. However, if the infection has reached the graft union, this method will not work. So the current options we have are either preventive or corrective, and they work from the outside in. So what we need is a method that we can work from inside out to equip the young vines with a microbiome that can help them stay healthy under high disease pressure. And we're best to find these microorganisms to help these vines. Well, we could look at vines that are already doing exactly this in the vineyard, and that is where we have the disease escape concept come into place. So the disease escape concept comes from the observation of some plants in highly infested vineyards, orchards, where these plants remain healthy when others around them are coming down with disease. As I use this disease escape in this presentation, I use it strictly as in pure epidemiological sense. That's not in a sense that they're genetically resistant to this disease, but they're genetically susceptible to the infection, but they're not just coming down with the disease. So as it relates to grapevines, usually in commercial vineyards, the vines planted are of the similar genotype, and they are also managed under similar management practices. So if there is a difference in the disease outcome, it points to a likelihood that the microbial community is involved. So this disease escape model has been studied in grapevines out of California in the context of the persistent disease of grapevine, and as well as in the oglobin disease of citrus. So in this research, the questions we wanted to answer are that which are the taxa that are present in healthy and diseased vines, and which of those taxa dominate in this group, and which GTD pathogens are mostly abundant in these samples, and which of these taxa discriminate best between healthy and diseased vines. Now, to answer these questions and more, I looked at 3 wine-growing regions in New Zealand, 1 in the North Island in Hawke's Bay, and 2 in the South Island in Marlborough and Canterbury. And to designate a vine as either a disease escape vine, it had to be mature. It has to be mature because the symptoms of trunk disease usually take about 8 to 10 years for it to be visible. So we wanted to see older vines. And also it must be growing in a vineyard with high trunk disease pressure. And obviously it must be free of trunk disease symptoms. We also wanted to use some objective measures like the leaf chlorophyll and the canopy size. So in order to identify these trunk disease escape samples, we used 2 methods. The first was the GTD mapping strategy. This work was carried out by Dion and colleagues previously, where they've gone through individual vineyards and they've assessed individual vines for trunk disease symptoms. Each of these boxes represents a vine, and in each for each vine, it could have a trunk disease feature of interest. It could the vine could either be dead or retrunked or had a canker, or it could also be free of any trunk disease symptom. So I had this map graciously provided by Dion, and then I looked at them, and then I went into the vineyard for a ground truthing to see if these vines that were identified on the map actually met the criteria that we wanted. So we had this map in Hawke's Bay and Marlborough, but not in Canterbury. So in Canterbury, we dependent on growers' recommendations because growers are usually aware of the disease pressure in the vineyards. So this is an example of a candidate disease escape vine. On here, you can see the chlorotic foliage on this tree, on this one on the right as well, and also this young one. Young vines are usually planted to replace dead vines or diseased vines. But among these, you can see this healthy vine with the nice foliage and very full canopy. This is an example of what we consider a disease escape grapevine. Similarly, you can see here in this slide, Here you see the disease vines here. You can see this one. It's already half head, no productivity, low productivity on this one. And you can see that a new vine is already being trained to replace this one. And there's also a dead vine here. And over there on the left, there's also another infected grapevine. But in the midst of the scales, there is one healthy grapevine that was growing luxuriously. And so this is what we consider a classic example of trunk disease, disease escape grapevine. So following the identification of these candidate disease escape grapevines, I took trunk samples from them and carried out some microbial isolations and DNA metabarcoding, which targeting the cystinase and ITS for bacteria and fungi respectively, and then sequenced on an Illumina MiSeq-based platform. The data was processed through the Data2 pipeline and was resolved into amplicon sequence variants. So one of the major questions I wanted to answer from this work was that what are there in the grapevine trunk? So from the deceased samples, we could see clearly that Eutypa appears to be one of the dominant taxon here. And you can see it was consistent across all the vineyards where we sample, where we had more Eutypa in the disease groups and than in the disease escape ones. So this one is for a pool sample, and the one on the right for individual vineyards. The Vineyard 1 and Vineyard 2 are from the North Island in Hawke's Bay, and Vineyard 3 and Vineyard 4 are from the South Island in Canterbury. Well, at this point, I must say that even though we went to 3 regions for the disease escape identification, we were able to take samples from 2 regions because of COVID lockdown. So, but in the disease escape group, we see 4 taxons that were relatively more abundant. We have Alternaria, Oribasidium, and Cladosporium, as well as Sematosporium. Oribasidium is a well-known taxon for its antagonistic effect against several plant pathogens. What is interesting here is that Femonella, which is also associated with trunk disease, appear to be relatively more similar relative abundance in both groups. And you can see here that it is more dominant in the North Island vineyards, but very low in the South Island. In the bacteria community, we see immediately that Pseudomonas group stood out as relatively more abundant in the GTD escape samples. So, but when you look at individual vineyards, we realize that the trend was not really as clear as we saw when we look at the pooled sample. For instance, if you look at this Vineyard 1, you can see that Pseudomonas was truly relatively more abundant in the GTD escape samples. But if you look at Vineyard 2, we see that actually Pseudomonas was more abundant in the disease samples, as well as in the Vineyard 3. You can see the Pseudomonas was more abundant in the disease samples, whereas in Vineyard 4, it was relatively more abundant in the escape samples. We know that the Pseudomonas genus is a very big genus with several species. Some of them are pathogenic to plants, and some of them are antagonistic to plant pathogens, and some of them are harmless endophytes. So what we did was to look deeper into the Pseudomonas taxon, and then we identify the different ASVs that matched to Pseudomonas. And then we broke them down into individual features. So what we could see immediately was that there are different ASVs associated with the deceased and disease escape samples. We could see here, for instance, this ASV 7, which takes up about 46% in the disease samples. But when we look at it in the GTD escape sample, it's almost non-existent in the GTD escape trace. But when we look at it in the individual vineyards, we realize that this ASV is actually dominant in this one vineyard. Now, when we look at this ASV 9, which takes up about 56% in the GTD escape samples, it appears to be a good indicator of GTD escape. But when we look at it, we realize that it's only coming from one vineyard in Hawke's Bay. And then we have this AXV8 that appears to be of similar relative abundance in both groups. Now, the interesting ASV here is this ASV21, which was relatively more abundant in GTD escape samples across all the vineyards. We can see here, you can see in vineyard 1, it was relatively more abundant than in the disease sample. In vineyard 2, it was relatively more abundant, as well as vineyard 3 and 4. So we saw that this ASV, it was not a side effect. It was a good indicator of trunk disease escape across all the vineyards. And what this also show is that only a few Pseudomonas species or subspecies might be driving the trunk disease escape phenotype. And it has also showed us to be careful about classifying these results at the genus level and to look deeper when signals like this comes up. So apart from the relative abundance data, we also wanted to find out if there are discriminated taxa that we can use to differentiate between the trunk and the disease state and the GTD escape state. So we use the random forest analysis, which is a machine learning tool that can discriminate between taxa. So the mean decrease accuracy is the higher the confidence in the taxon as a discriminatory taxon. So we can see here in the fungal community, Utipa was the highest discriminatory taxon for the diseased group, whereas Orobasidium and Cladosporium were the two highest taxa that discriminates for the GTD escape community. We also had this Rhodotorula. And for the bacterial community, surprisingly, Hymenobacter appear to be the taxon that best discriminates between diseased states and the GTD escape samples. And also following that, we have Pseudomonas as the second most discriminatory taxon for trunk disease escape. So we also carried out some more analysis to differentiate between the states and to see which microbial taxa associate with these different states, which time will not permit me to discuss. So what these results provided for us is a short list of taxa that we can associate with the disease escape vines. Although they are candidate indicators of this vine health, they are not proof of causal protection. So what it provides for us is a basis for selecting isolates for inclusion in a synthetic microbial community. Because these days we are thinking towards using a synthetic microbial community to combat a complex disease like grapevine trunk disease, which is caused by different pathogens. It's unlikely to be one solution. So synthetic community aims to reduce the complexity of the natural microbiome and then to replicate the functions of different microorganisms and then use them as a protective microbiome for young grapevines. So we found out that disease escape grapevines can be found in New Zealand vineyards, and we've observed this and documented this. And we discovered that there are key fungal and bacterial taxa associated with the trunk disease escape phenotype in New Zealand. And Pseudomonas appear to be the clearest and the most consistent taxon associated with the disease escape phenotype. However, the question still remains, can these microorganisms protect grapevines against the negative effects of these trunk disease pathogens? Well, the answer lies in future work where the synthetic microbial community will be tested to see if they can establish in grapevines and protect against trunk disease pathogens. So if you're interested in this work, you can scan this QR code to learn more about what we found and to see more of the analysis we carried out.
Thank you
I would
very
like to
much.
acknowledge my supervisors and people who have contributed to this, and Bioeconomy Science Institute, Lincoln University, and MB for the funding, and to all the vineyard managers across our sites for providing us access into the vineyards to take samples and to conduct our survey. And thank you very much for listening to the presentation. Thank you, Joe.
Well, thank you very much for the presentation. Great work. And,
Yeah,
as you showed at the end, you need quite a number of people to carry those very large experiments and
yeah,
large project, but at the end, very exciting.
yeah,
So if anybody has a question, please write it in the chat. We have perhaps, just time for one or two short questions. I do not see anything in the chat, so I'll have a quick question, Damola. You talk about synthetic community. Are
yeah.
you thinking about mixtures of fungi and bacteria, or are you more focusing, for example, on different ASV in the Pseudomonas group, or and how you're going to determine what synthetic community would be the most appropriate? Is it based on the effect of single strands, or are you going to bring together a group based on the taxonomic plot you showed, or yeah?
Yeah, so we looked at different combinations of the synthetic microbial community. So we looked at putting the bacteria together, and we also looked at putting the fungi separately, and we also look at the combination of the bacteria and fungi. So we used different methods to select the final candidates. We wanted to see, for example, if they have in vitro antagonism against trunk disease pathogens. And we also selected them based on their association with the disease escape phenotype. And because they don't have in vitro antagonism does not mean they're not a good candidate for inclusion in the synthetic community. So they could be for example, the ketone taxa that brings all the community together. So the most important thing we wanted for the selection was the association with disease escape phenotype from our bioinformatic analysis. Yep.
All right.
Yep.
Okay, thank you very much. Great
Yeah.
presentation,
Yeah.
great work, and all the best for the future, because
Yeah.
this project will keep moving forward, I'm sure, and
Yeah.
we'll hear later
Yep.
from you and the project.
Yeah.
All right,
Thank you.
time to move to the second seminar, the second presentation of the day, and that will be given by Monica Gerth, who is from Victoria University of Wellington, and she'll be talking about a very important group of pathogen, the oomycetes, and in particular, how to detect them and identify them. So Monica, please, anytime. And we see your slide, so it's perfect.
Excellent. Whoops. Can you still see it?
Yes, yes. And
Okay,
thanks, by the way, for accepting the invitation to come to talk today to us. Really appreciate
No, thank you very much for
Thanks.
having
Yeah.
me.
Yeah.
So as Joel said today, I'm going to talk about a new test that we've developed for detecting Phytophthora, in particular Phytophthora agathodicida. My talk today will have two parts. I will warn you, part one is most of it, where we will talk about how the test works and how we've validated it, and then a very short bit, if I have time at the end, to talk about an application, so a way we think it could be useful. And before I launch into the science, I just want to do the acknowledgements. I want to thank my lab group, past and present. I'm based at Victoria University in Wellington, and most of the work that I'll be talking about today was started by Jade Palmer, a very talented PhD student. Emily Hawking has been working with Tiakina Cowdy to do lots of the validation, and then particularly Izzy, I think I'll talk a little bit about her work at the end, and our collaborators, the Tedora Cowdy Audit team, who have helped us make sure that our diagnostic is fit for purpose and something that people can actually use. And of course, all of the funders and everyone that has shared time and data with us. So Phytophthora, I think for this crowd, I don't really need to introduce the genus, but in terms of New Zealand, one of the most famous Phytophtheras is Phytophthora agathodicida, which has been causing root rot and dieback disease in our kauri trees since at least the 70s, possibly longer. It causes these very characteristic symptoms where you get bleeding lesions, collar rot, and ultimately tree death. And kauri dieback disease kills kauri of all ages, which of course in New Zealand is very problematic because these trees are culturally, economically, and ecologically very important. In terms of tools, right now, accurate surveillance is really the key to management. You need to know where the dieback pathogen is so you can close tracks, install hygiene stations, and guide things like phosphite treatment. In New Zealand, like most parts of the world, one of the main tools for surveillance is still baiting. So currently, all approved detection methods for agathodicida rely on soil baiting. So baiting is a very well-established method where you basically fish for the pathogen using plant material as the bait. It takes relatively large amounts of soil, so about 250 grams per sample. You go through multiple steps of drying, baiting, and then you can either use a DNA-based method, such as PCR or LAMP, or you can culture the pathogen and identify it morphologically. And these are the two methods that are approved in New Zealand. So baiting followed by DNA tests, or baiting followed by morphology. There have been attempts to make a DNA-based test work directly on soils, but there's never been any that have been sort of sensitive enough or reproducible enough. The problem is there are some limitations to baiting-based methods. It's slow. So for PA, it takes about 25 days. It takes a relatively, as I said, large amount of soil. And because in New Zealand, Agathodicida is an unwanted organism, baiting has to be done in an improved containment facility. So this makes the baiting test more costly, about $200 per sample. And of course, it's just inherent for baiting that it's not quantitative. So you get a yes/no as to whether or not the pathogen is present, but you don't necessarily know how much is there. And I think particularly for New Zealand, it can be a little bit problematic because for baiting of Agathodicida, there is a tendency towards having high rates of false negatives. So we have a lot of other competing Phytophthora species like Cinnamomi that are present in our soils, and they can outcompete Agathodicida in the baiting process. So we wanted a method that would get around some of these constraints. And the O in our ODNA test is for oospore. So oospores are a really important part of the Phytophthora Agathodicida life cycle. PA is homothallic, so it produces oospores very readily. They're released into the soil by infected trees as the roots die back. And once they're in the soil, they're very tough. So these are the survival spores of the microorganism, and they can survive in the soil for years. Once they're there, they don't move around on their own, but they can be spread by movement of infested soils by shoes, wild animals, livestock, or other equipment. So the O in the ODNA test, the O is for oospore, and that is our molecular target. And our wish list for detection method was we wanted something that could still provide that quick, easy yes/no answer, but we also wanted quantitative data. So the easiest way to meet both of these would be DNA-based tests. And we've had those for lots of other things. It's really huge for environmental monitoring, eDNA-based monitoring. And also, of course, we all know about DNA and RNA-based tests for human health pathogens. So why haven't we actually had a DNA test that works well for cowdy dieback disease? And I think particularly for Phytophthora and definitely Phytophthora Agathodicida, there's a few challenges. One, as I said, the DNA test has to be highly specific for PA and not all of those other Phytophthora species that are in the soil. In the soil, the oospores that we're targeting are present in low amounts and heterogeneously scattered, and that's why typically baiting you have to have around 250 grams of soil. And as most people will know, 250 grams is not really compatible with most DNA extraction kits. So most standard kits only handle about 0.25 grams, and the soil itself can inhibit the DNA tests. And then the final, I guess, hurdle that we faced was that if we got the oospores, it's actually surprisingly difficult to get the DNA out. So DNA in Phytophthora oospores is encased in those hard to break spores, and I'll show you that in a minute. So I won't talk about this too much, but basically we were lucky enough to be collaborating with Rosie Bradshaw at Massey University on the PA genome, and we got early access to it and were able to find a region, a new barcode region in the genome that repeats over 470 times in the genome. So that gives us great sensitivity for our test. And bioinformatics screening suggested that the sequence wasn't present in other species. So hopefully strong specificity. In terms of the sensitivity, we found that with our endpoint PCR version, so this is the quick and friendly yes/no version, we get reliable detection down to 0.01 picograms or 1 femtogram. And for the quantitative PCR version, the one that'll give us that information on pathogen loads, we have a limit of detection of 0.7 femtograms, so basically still 1, and a limit of quantification of also 1. So this is very highly sensitive. In terms of the specificity, I will show you a lot of ugly tables, but I'll whiz through them. The first question, of course, is, does this new barcode region actually exist in all of the different Agathodicida isolates? So we tested 13 geographically diverse Agathodicida isolates, and all of them test positive using our PCR assay. So that was great. The other question, of course, is this actually specific? And so we also tested genomic DNA of 23 related soil microbes, mostly Phytophthora species, but we also tested some Pythium and some other ones. And so it was 13 shown here, plus an additional 10 more. And all 23 of the related soil Phytophthora and other microbes were negative in our PCR assay. So we're detecting all of the PA we want to detect, and we are not picking up all of the related species that are present in the soils. I do want to point out that there have been other primers developed for Phytophthora agathodicida detection, and our new primers have improved specificity. So the previous ones were also non-specific in that they pick up other clade 5 Phytophthoras and also can cross-react with Cinnamomi, which, as I said, is really common in our soils. So we're pretty happy with the sensitivity and the specificity with genomic DNA. But of course, we're not actually trying to analyze genomic DNA. We're trying to get actual oospores from soil. And so we had to tackle these other two challenges. And the first one, which is I think the one that's probably most broadly applicable to other organisms, would be our teabag method for filtering the oospores away from soil. So we did trial lots of different methods. And what we came up with was shown here, 2 different tea bags with different pore sizes that can enrich or concentrate the oospores from soil. So the inner tea bag holds roughly 10 grams worth of soil. This one has large pores that trap the soil particles, but let everything smaller than 90 microns through, that includes oospores. And then we put this inner tea bag inside an outer tea bag, which has smaller pores. The smaller pores trap the oospores while letting everything else pass through. So everything smaller than an oospore passes through to the bottom. All of those inhibitors that are present in soil get washed away, and you can recover the resulting oospore slurry from the outer bag and then go through a DNA extraction protocol. And so we think there are a lot of advantages to this method. So it allows larger sample volumes to be used with standard DNA extraction kits. So as I said, we start with about 10 grams instead of 0.25. It was also really important to us that it's low tech, low cost, and no cross-contamination between samples. So molecular techniques are often prone to having false positives popping up from contamination in the lab. And the tea bags are one use, you throw them away and you get a new one. And as I said, the ability to filter away the soil also reduces the presence of all those pesky PCR inhibitors present in soil, as well as competing DNA from non-target organisms that might interfere with our PCR reaction. So we now had better primers. We had a better way of separating the spores away from the soil. And it was really interesting that when we put our oospores through a standard Power Soil Pro kit, what we found was shown here in that these are oospores, and hopefully you can see they're largely intact. So what we found was the oospores of agathodicida pass through a standard DNA extraction kit intact, which means you're not getting the DNA out, and if it's not outside of the spore, you can't amplify it. And what we found was after much trial and error, we could just substitute in the bead beading tubes with an optimized mix that breaks open. We estimate, this is the lysed oospores, about 98%. So this makes the DNA accessible for PCR. We found it definitely needed a very specific mix of glass and ceramic beads at 3 different sizes to work effectively. We tried lots of different versions, and if we varied any one variable of that, it did not work properly. And so this is just an overview of the full method. You start with 10 grams of soil, you place it in your double tea bag, you leave it to soak in water. The inner bag with the soil is discarded. The outer bag is rinsed to collect the oospores. We can then take that pellet and process it with our new improved lysis and primers in either PCR or qPCR. And so we're really excited about getting this into the field. And so for the last year or so, since we originally published our method, we've been going through a process led by Tiakina Kauri, who have assembled an independent advisory group. And the focus of the group has been on interlab reliability. So is the detection limit that we find of 1 femtogram consistent across different labs and sample types? And also, if we apply this to soil samples, do different labs, operators, and equipment, do they get the same results? And so we started off just working with pure DNA, but at different concentrations. So we had a panel of DNA samples going from above and to below the detection limit, and also in water backgrounds and eDNA backgrounds, again, to sort of probe the specificity. We prepared blinded DNA panels sent to 2 independent labs, and this was our first really exciting result. So shown in green, if it's green, it means it's matched across all 3 labs. So our lab and 2 independent labs got the same result across the range of concentrations and sample types. And then, of course, the much sterner test is blinded soil samples. So we also assembled a panel of 20 blinded soil samples sent to the same 2 independent labs. These included 10 known PA-positive samples, so these were symptomatic trees that were baiting positive historically, and 10 PA undetected samples. So these were ones that were historically baiting negative, and we've also pre-screened them to make sure. And this panel was assembled to both have a range of the positive and negative samples, but also a range of concentrations. So when we pre-screened the positive samples, it was a panel that ranged from about 4 to 400 femtograms of PA DNA per gram of soil. And we gave the labs training and protocols. There were some variations to our protocol due to supply chain issues, but other than that, it was pretty uniform. And this is the results. Our results are in the first column, 10 positive, 10 negative, and then these are the interlab results. And what you can see is 9 of 10 for each independent lab were positive of the 10, or 28 out of 30 replicates total. We had 2 false negatives due to potentially soil heterogeneity, possibly operator inexperience, or it could be substitution of the different extraction beads. In terms of the negative samples, across all 3 labs, so 10 times 3, 28 of the 30 were in agreement, 2 were inconclusive due to extraction control failures, and I think under a normal situation, you would do some troubleshooting and rerun them, but this was, for the interlab testing, that wasn't an option. But overall, we were very excited by this. This was a very high rate of agreement. And just for comparison, the reliability of the approved baiting and/or qPCR methods, it's always been problematic, the reliability across labs. So a 2014 study looked at the interlab reliability using 25 known positive trees and found very low rates of detection or agreement between the labs via baiting, where one lab found 3 out of 25 positive and the other one found 7, or via qPCR, where one lab found 4 out of 25 positive and the other one 9. And so these were 25 known positive trees. So these are relatively low detection rates. Subsequent studies have shown that the optimized baiting protocols in New Zealand achieve an estimated detection rate of 63%. So if you have 100 positive trees, you'll detect PA 63% of the time. And we think in comparison, the 90% detection we're seeing is therefore hopefully a bit of an improvement. So beyond the blinded soil panels, we have started testing wider panels of field samples. So we've also tested 65 samples. This was just our lab now, not the other interlabs, that were collected and previously tested using a hybrid baiting lamp method as part of routine surveillance. Our new and improved method detected PA in 45 of the 65 samples from kauri forests, whereas baiting lamp method only detected PA in 7 of the 65. So we think this reflects the higher sensitivity of our assay. And we do see this when we look more broadly. So we find frequent detections of PA in kauri forests with symptomatic trees. On average, we've done about 168 samples now, and we're getting PA detection 68% of the time. In many cases, it's low levels around healthy trees. Sick trees have much higher levels. I'll show you that in a minute. I would say in terms of specificity, we've never detected PA outside of kauri forests. So we've tested Wellington kauri, we've tested potting mix, we've done a national survey of the non-kauri growing regions, and they're all coming up negative. So We're pretty confident, given the specificity testing, plus these other known negative samples still being negative, that what we are detecting are true positives. So for the part one summary, I guess the big picture points are, I think we've got a test that is now more sensitive and specific than existing methods. We've got improved interlab reliability. It's a fast method. You can process multiple samples from start to finish in 1 to 2 days. It's more affordable. So compared to baiting, which is currently over $200, we estimate our total cost of our test is about $85, which is $30 worth of reagents and then some money for time and overheads. And a really big part for us was also that it's more accessible. So you can use relatively simple equipment, cheap PCR machines, which opens up the possibility for community labs and on-site testing because there's no culturing involved anymore. It doesn't have to be done in containment. It also has the advantage that less soil needs to be collected and disposed of. And what I think we're most excited about with this is we just get more data. So the ability to have quantitative data and actually see trends over time and disease symptoms, we think will be really powerful for guiding sampling, identifying hotspots, identifying the impacts of attempted control measures, disease progression, and more. So our original paper was published a year ago, and that's the URL for it. And now, in my last couple minutes, I just want to show an example of how we're using the test to start optimizing some of the sampling strategies and also just answering other research questions. So when we first went out actually with Tororo to do some sampling, I remember standing there and being like, oh yeah, no, you don't have to dig such a big hole. We don't need so much soil. And they rightfully asked me, well, do I just need to dig the same depth hole and take a smaller amount of soil out, or can I dig a smaller hole and disturb fewer roots? And they helped us test this. So they sampled 8 trees from Waipoa Forest at 2 different depths. So this was a very, I guess, field realistic sampling. This was using trowels, not soil cores, but they basically dug shallow holes next to the trees and collected the smaller amount of soil from just the top 10 centimeters, or deeper holes, which is their standard depth, collect down to 20 centimeters. And in every case in this pilot study, all of the positive samples, which in this was 5 of 8, the shallow soil samples contained higher concentrations of PA-DNA than the deep samples. And that's what's shown here. So the shallow, the amount of PA-DNA in orange, and then deep was in green. And just as a reference, 1 femtogram of PA-DNA is roughly speaking 1 oospore. So we thought this was really promising, that shallow sampling, you could dig smaller holes, you disturb the roots less, you have less soil to carry out and less biohazard soil to be disposed of. But this was obviously relatively crude in that we weren't looking at the abundance at different depths very carefully. So we did go through and do an additional 12 calorie trees over a range of disease symptoms where we took 4 cores per tree. Each core was split into 4 layers, so roughly 5 centimeter increments. Each depth was pooled into a single sample, and then we processed all of these 48 samples using our quantitative PCR method. We have a preprint of this online. I just want to point out here, so these are the non-symptomatic trees, possibly symptomatic and symptomatic. And what I think is really exciting is that we're also seeing not just differences with depth, but we're also seeing really strong differences in abundance with symptomology. And so non-symptomatic trees tend to have low levels of PA present, typically in the shallow depths. Possibly symptomatic, we go from about 5 to 100, and then with symptomatic trees, we're up above 250 and into the thousands. And so it basically starts shallow and not very abundant, and as it replicates, it goes deeper and increases. And so we think this is a model where PA is getting tracked in, probably by people's shoes or wild animals. It can be found around healthy trees, but in very low abundance. Then as the trees are infected, they start shedding large amounts of oospores into the soil, resulting in high levels of PA at deeper depths. And we think this is a nice example of how the quantitative version of our test can be used to actually start quantifying PA abundance in soils and hopefully improve management. And so in my final thoughts, I just want to say, I think in terms of baiting, particularly for Agathodicida, I think we have some advantages in that it's not confounded or not outcompeted by other Phytophthora species, and it provides quantitative data. And I also think compared to traditional eDNA type studies, I think it enriches for oospores over free relic DNA, and oospores are an infectious propagule, and it also allows for an increased sample size. In terms of other applications, we think the filter bag method is adaptable to other systems. You can vary the pore sizes to include or exclude different size organisms of interest. If you have a microbe or a nematode, you can probably adjust these. We find 10 grams works really well for our system, but you can also vary the tea bag volume to optimize for larger or smaller sample sizes. And what we're finding in metagenomic studies, it's still early days, but we're finding improved diversity captured. We've been doing some work with raspberry growers and Tamarilla growers, and we're getting much better results in terms of the captured diversity by using these larger soil sizes via the tea bag method. And so, yeah, that was it. I am happy. I just want to thank everybody again, but I did that at the start. And so, yeah, I am happy to answer questions.
Wonderful presentation, Mika. Very exciting. Your results are fantastic, and I really like, the idea of the tea bags. It's wonderful. It's beautiful. Now we're slightly out of time, but there are 3 questions. If you could answer those questions relatively rapidly, because as I said, we're already slightly out of time. So the first question is: did you compare detection method with the baiting method developed by PFR-BSI in Hawke's Bay? So I'm not quite sure what baiting method they're talking about, but you might know what they're talking about.
Yes and no. So all of our samples have been baited, but our results for that are historical bait results.
Okay.
We do actually think I don't have time to talk about it today. I'm happy to talk offline. We do think there is more of a difference between the LAMP assay and the morphology-based assay than maybe people thought.
There's a question from David. Perhaps you can read the question so people who don't see the
sorry.
chat can
One was
hear
how
it.
tools are cleaned between tree sample collection. And we use bleach. We recommend bleach.
Yeah.
I'm going to jump to the easier ones first. CRISPR-based, we've definitely thought about it. People are asking if we've thought about CRISPR-based. I think there's lots of other optimization we could do, but our focus at this point is trying to get it approved and out into the world, and then trying to do more, refine it to make it even easier. And then another question from Louise: do we know if our identified barcoding gene region is variable and specific in other Phytophthora species? We think ours is specific in PA. We've never found it in anything else bioinformatically or testing against genomic DNA. So yeah, we think the rest of our method will be adaptable, but the primer is probably not much use to anyone else. And then there was one more from David asking, given Phytophthora develops exponential epidemics, the initial level of inoculum is less important than environmental conditions that favor infective zoospore production and release. Can you explain why measuring pathogen load oospores was a priority and how it helps preventative management? I guess in terms of priority, for PA, baiting is basically, the current baiting protocols are based on oospores. So they dry the soil to disfavor things like cinnamomi. And so that's one reason. And yeah, I mean, the zoospores will be produced from the oospores germinating. And so the oospores are the longer lived propagule. And so that's why we went after them.
And while you were answering, another question came very quickly about it, because I see we're out of time. How about combining your spore collection and DNA extraction method with a LAMP assay?
Yeah, we did actually. I mean, I think in principle it should work. And if you wanted to do it with a different Phytophthora species, we had dreams in the beginning of combining our extraction method with a colorimetric lamp, again, to make it more user-friendly. We were never able to reproduce the detection limit of the published lamp assay, so we're not sure what went wrong there. But yeah, that's why we switched back to other methods like qPCR.
Yeah. All right. No, very good. And I guess if people have more questions, they can contact you directly and to be in contact with you about,
Yeah,
whatever
definitely.
what he wants to hear, and the same for Damola. So anyway, I think it's time to wrap up. So Noria Kina Kai Korero, Namihi Nui Kia Korua. Thank you very much to the two speakers. I mean, fantastic that you shared your work and your experience. And of course, Namihi Kia Kotu Katoa Itai Mai. Thank you to all the people who came. I've seen quite a number of people assisted. At the end, the seminars are for all the people, and it's great to see people attending the seminars live. And Kotu Kutu Kamanako, Kakite Yakotu Itemarama Itumai. I hope to see everybody coming back next month for the next seminar. As I said, those APPS seminar are a monthly occurrence, and it'd be great to see all of you again next month. And in the meantime, take care. Matewa. Bye.